Hi all, welcome back to Microbiology Lectures. Today we're going to talk about some of those more niche techniques that are important for certain industries. Some of them maybe you haven't heard of or might not be as familiar with. But again, lots of industries the goal of those industries is to prevent microorganisms from growing or destroy the microorganisms that are growing. Okay, and one technique that's fairly old, I think the FDA used it first in the 1970s. It's been used a lot more, but to treat food, irradiation was used, I think, in the 1970s to treat flour, so wheat and flour from wheat. But anyways, let's kind of dive into why it works or kind of how it works. Okay. Electromagnetic radiation, we know it as the waves that are coming from the sun, or we can recreate these waves using electricity. But the waves that are coming from the sun can be in lots of different wave patterns or wavelengths, and the shorter the wavelength, the more energy that wave gives off. Okay? And so the visible light spectrum, where we're most familiar with these waves because this is kind of how we see, sits at about 390 to 750, 60, somewhere in there, nanometers. Okay? Anything really from about 300 nanometers and lower will often be considered or have germicidal properties, which means they can be used to destroy living tissue, germs or pathogens, viruses, bacteria, protozoans, fungi, those kind of things. And we've been using these for a very, very long time, especially ultraviolet light, but now more recently we've been utilizing X-ray and gamma rays because they're more potent, they can travel further, and they destroy more than just what UV does. So ultraviolet, because we've known about it for quite a while, and the dangers of ultraviolet light to humans, from like the premise of skin cancer, or even cancer in other organisms, like plants can get cancer from too much ultraviolet light and other things. Because what ultraviolet does when it interacts or penetrates a tissue, it can interact with DNA. And what can happen is it causes the DNA to form a dimer. A dimer means that instead of bonding across, nitrogenous bases bonding across, they bond on the same side. So they bond on the same strand of DNA, and they form a bond together, which causes the DNA to pinch. And when that's replicated, often that replication results in a different reading code, a different messenger RNA, and therefore the protein that's generated from that code is not the same. And if this was a coding region where that pinch or that dimer forms, then that can be dangerous to that organism. Okay? And that's kind of the premise behind the ultraviolet, or use of ultraviolet. Gamma rays, on the other hand, do that, and they create free radicals, and they can damage the cytoplasmic membrane. So ionization or ionizing radiation, that's gamma rays and X-rays, they'll typically destroy the DNA by causing dimers, okay, but then also disrupt the cytoplasmic membrane and produce free radicals, so they can cause oxygen to be a more reactive species. They can take water and get the oxygen from the water, but also oxygen from the atmosphere, create free radicals. And we talked a little bit about free radicals and how this works. When you create these free radicals, they're very reactive. Okay, so they'll bind to different molecules, and what we often see is they bind up to proteins. And when they bind to proteins, it causes the protein to change its shape. And when it causes that protein to change its shape, then maybe active sites or allosteric sites, these other sites that are on the protein, that, like an enzyme, that are needed to be a specific shape, they're no longer that shape. And that causes irreversible damage to the organism. And so we've been using high energy gamma rays for quite some time now. This is in some cases, this is a sensitive subject to people, because we know what gamma rays, X-rays, UV radiation can do to the human body. We know it can induce cancer. We know that it can destroy the DNA and cause mutations. Yet we've been treating food products with it for quite some time. And so we know that irradiated food lasts longer, has a longer shelf life than non-irradiated food. And so you can see some strawberries here that were not irradiated and some that were. Okay, this works on a couple profiles. Okay, so first of all, when we irradiate the food, it destroys, the DNA, the cytoplasmic membranes, create free radicals, and most of the organisms that could be considered spoilage organisms are gone. Okay, and so that increases the shelf life of the organism from a non-pathogenic perspective. Okay, the other way at which we can treat organisms is shown here, and that is we can stop the organism itself from producing tissue. So going to seed. We can, use ionizing radiation to prevent that living tissue from generating. And so you can increase the shelf life that way. Problems with it, of course, it can cause damage to cell walls and membranes in the tissue if not done correctly. So it can cause malformation in the tissue, can cause some damage that way. But like I said, really, I think it's perspective of the consumer and how these different products are treated. And some people have an issue with the use of high energy to treat food and rid it of spoilage organisms, and other people don't. Okay? But one way in which we use ultraviolet radiation in tons and tons and tons of industries, and many water treatment plants or water treatment facilities use ultraviolet to treat the water. It works really well to destroy microorganisms or microbes in non-turbid solutions. And so water is a great one for this. So after the water has been filtered, we talked about filtration before. After it's been filtered, it then can be treated by using UV radiation. The problem with UV radiation is it doesn't have the penetration power that gamma rays do. And so gamma is much better at penetrating through material, through packaging material, through turbid material, so things that have a lot of particulate matter in them, so like fruit juices and things like that, is much better at penetrating those. UV tends to not penetrate very well. It doesn't even really penetrate plastics or glass, but that makes it even more important for the safety of the individuals involved. So it's typically considered much cheaper than gamma radiation. Is also typically considered much safer. So for example, lots of water treatment facilities, After they've filtered, they run the water through these UV tubes, and so inside these tubes are UV lights, very similar to this handheld light, but it's a UV light that's in these tubes, and the water just runs past it, and the UV radiation zaps microorganisms that might be still in the water. The nice thing is individuals don't have to use or have specialized PPE or personal protective equipment to work these facilities, because the UV is blocked by the pipe or the PVC, and they don't have to worry about damaging their skin or their body by working at these facilities. In the gamma situation, it's much more controlled, because those gamma rays can penetrate through a lot of the equipment, a lot of the material can reach, or gamma rays can penetrate through. Okay? So much that UV is considered safe enough that individual consumers can use these UV products to treat their own water, like if they're on a hike or whatnot, and they've taken water from a stream or something, they can treat it. All right, high pressure is another technique. So high pressure processing, it's used to be called cold pasteurization. I think now pascalation is more of a popular term, but all of them are used interchangeably, and the process is really no heat. So this is the nice thing about this process, is instead of using heat, you're using pressure. So the food will go into a pressure chamber, and the pressure will be increased. So, sometimes around 120,000 pounds per square inch, depending on the microorganisms, also depending on the product that we're talking about. It works really well on fruits and veggies that don't have air spaces, that have a good like rind, so like oranges and lemons and tangerines and these things that have a good rind, not too much airspace inside. It works fairly well on those to kill the microbes that might be occurring on the outside of the fruit or veggie. It also works well on human-made products like guacamole and salsa and things like that are often done. Fruit juices, another thing that's commonly done. It will destroy the microbes by denaturing their proteins, so it causes the protein linkages, the peptide bonds, to break or twist, and then it changes the shape of the protein, and it's not reversible. So the problem sometimes with this is, because it's denaturing proteins, whatever the substance is, whatever the material is that you might be using this on, if it's protein based, it could denature enough proteins to change the flavor. But again, the idea is utilize this to maintain the color, the flavor, with fresh food, and so a lot of fruits can be utilized for a lot of fruits. Other non-suitable foods are things like dry foods, or definitely things with air pockets, so like bread and stuff like that have these air cells, from carbon dioxide during the bread making process. Those are like little chambers that they want to swell too, and they tend to destroy the product. So again, denatures proteins. That's great, unless the protein profile of the product you're after is fragile. But the other thing it doesn't really do very well is eliminate viruses, which can be problematic. Okay, and then probably the one that most people are familiar with is the utilization of chemicals. And so chemical methods to destroy microorganisms is everywhere. Okay, probably more so than ever in the past, with the most recent kind of outbreak of COVID. Huge amounts of chemicals are being used to disinfect, in an attempt to sterilize regions, to stop the spread of not just COVID, but the flu or any bacterial because quite frankly, you know, we were laxadative until something happens, and that happens a lot. And we'll talk more about kind of society and how we go about things. Like when there's an outbreak, whether that be foodborne illnesses or whether that be, you know, a new flu that comes through and has a higher virulence, or you know, something like Zika or something that might be insect carried. Until those things happen, we often are much more lax, and our procedures go down. We don't clean as much. Those kind of. Then something happens, and then everybody's back up on high alert. And so germicides are everywhere now. So sometimes the germicidals can sterilize, and we talked about this a little bit before. The idea with a germicidal, one that can be used as a sterilant, is that you would use this germicide in conjunction with multiple things. So multiple products or multiple ways to treat things would come into one kind of cocktail. And so you might have things that are attacking proteins, denaturing proteins, destroying DNA, destroying the membranes or making the membranes more loose and able for more product to get in, destroying viral envelopes. So lots of ways. And so a typical product is a product that will have multiple ingredients that might go about doing these things, and so we consider them cocktails of chemicals. Because of the cocktail chemicals, sometimes we don't know the actual mechanism by which it's destroying that virus or that bacteria or that fungi. And so sometimes it's poorly understood. The other thing that is sometimes poorly understood is what is meant by the labeling. So in the case of things like Lysol, and there's lots of other products out there, they'll often label things like kills 99% of germs on kinds of surfaces, all kinds of surfaces. Okay, just so you know, when we're talking about products that have these claims, I'm not discrediting their claim. Okay, I want to make that to be sure you understand that. What I am telling you though is it's 99.9% of the microbes they tested. Okay, so novel microbes or things that they didn't test, they don't know that it actually kills them. Okay, and that should be like common knowledge, but I think most people, when they pick up a product and it says it kills 99.9% of germs, then it's going to kill whatever you're worried about. And in some cases it might, in other cases it may not, because it's never been tested on that. Okay, so keep that in mind. The other thing that I want to talk about when we talk about germicides, these are chemicals. Chemicals are typically worse. They do not work as well as temperature. So like we talked before, autoclaving is like the go-to if you're going to sterilize something. The nice thing about chemicals is they can be used on all kinds of objects, doorknobs, desks, toilets, whatever it might be, things that you cannot autoclave. But they're less reliable. So because of how they're applied, also because of the way they work, they don't do as well of a job at sterilization as things like high temperature, high pressure things. Okay. The other cool thing about though the germicides is that depending on the germicidal properties of that chemical, Some of them are safe or non-toxic enough that they can be used as antiseptics and prevent the growth on living tissue. So remember, an antiseptic is going to be applied to living tissue, whether that be human or a vet in treating animals and those kind of things, but living tissue. Okay, so when we come back, I'll talk about the categories of germicides and kind of some specific examples on the usefulness of different germicides.